Good morning, good afternoon, or good evening. No matter where you are in the world, we warmly welcome you to our latest Novogene webinar. Today, we are delighted to introduce Thea Zieglar, Sequencing Application Sales Specialist at PacBio. Thea will be presenting today's webinar, looking at how long-read sequencing is opening a new era of genomics. Thea will be joined by our Deputy Product Director, Shi-qi Ann. For those of you who are new to our webinars, you can find on-demand recordings of all our previous webinars on our website. These include our ever-popular beginner's guides, webinars which introduce the basics of RNA sequencing, whole genome sequencing, microbial, and a lot more. We'll send you a link to find these in the follow-up email after this webinar. A bit of housekeeping before we start. The presentation will run for around 40 minutes, with 15 minutes for questions at the end. We encourage you to submit your questions using the Q&A feature located at the bottom of your screen. You can upvote a question by clicking the thumbs up symbol next to it. If we don't have time to answer your question live, we will be in touch via email after the webinar. This can take a few days though, depending on how many questions we have, so please be patient. Please don't use the raise hand functionality. For any questions or if you encounter technical difficulties, please reach out through the Q&A section or message us directly. We'll share the slide deck and a recording of the webinar via email. This should be with you on Friday or early next week. If you are studying and need a certificate of attendance, there'll be a link in the follow-up email that will display a certificate you can print directly from your browser. Finally, there'll be a short survey that pops up when the webinar finishes. Please take a couple of minutes to complete this, as your feedback is invaluable to us and help the content of future webinars. That's it from me. I'll hand over to Shi-qi Ann, who will be hosting the webinar.
Hello. Yeah, let me just
Hello,
set up.
Shi-qi, I think you're on. I'm going to pop backstage then.
Okay, thank you very much. Just bear with me. Let me set up the slides for a second. And I just wanted to say thank you again for joining us today for our joint webinar that is between PacBio and Novogene. And the topic is on long-read sequencing, the next era of genomics. Okay, so I'm Shi-qi Ann, and I'm the Deputy Director of Product Marketing at Novogene. I'm delighted to be your moderator for this session. And also we're thrilled to have you today and to dive into the recent achievements and applications of long-read technology. Okay, so join us today, as John already mentioned, we have special guests from PacBio. Dr. Thea Zieglar and Dr. Dimitar Petrov. I will tell a little bit more about them later. Okay. So let me start by introducing Novogene a bit. So Novogene founded in 2011 and has grown into a leading global genomics service provider. So our state-of-the-art laboratories and sequencing facilities span multiple continents, as you can see from the map here. With over 2,000 employees worldwide, including leading experts in genomics and bioinformatics, we're committed to advancing genomic science. And also in Europe, Novogene operates state-of-the-art laboratories located in Cambridge, UK, and also Munich and Germany. And our dedicated local team provide comprehensive support and expertise, ensure we serve our European customers with exceptional efficiency and precision. Okay. And next, I want to take a look at our journey with PacBio. So Novogene, as I mentioned, founded in 2011. And since then, we have actually integrated several PacBio systems. So in 2017, we integrated the SQL system. And then in 2021, we added the SQL 2E systems. And by 2023, we actually expanded our operations globally. So with the 6 PacBio Revio system globally. And we launched, that's actually include also our German lab as well. And 2023, our German lab was founded as well. Okay, and then in early this year, we actually launched the PacBio Kinnex-based full-length 16S sequencing and also Kinnex-based full-length RNA sequencing services as well. So you're going to hear a little bit more from Thea later. And also we're very proud of our achievement with PacBio technology. So we have processed approximately 10,000 samples globally for PacBio sequencing, and we contributed to construction of reference genomes for over 1,000 species as well. So since launch, this is very impressive. Since launch, we already run over 2,000 PacBio Revio smart cells. And actually we achieved a very impressive PacBio Revio HiFi data output record as over 135 GB per smart cell. And in 2023 alone, we facilitated 140 publications with long-read sequencing and with accumulated an impact factor over 1,070. So as you can imagine, Novogene offers a wide range of PacBio sequencing services, so including everything listed here, including whole genome sequencing. This includes de novo sampling, can work with standard input and ultra-low input samples, and also microbial de novo genome sequencing, and for recurrent sequencing or for variant detection. And RNA sequencing, we offer a full-length RNA sequence. This is a 4-bark RNA-seq, and it is including standard iso-seq and also Kinnex-based applications as well. And for single cell at the moment, we offer the pre-made library sequencing services. And so for metagenomics sequencing, we offer full-length 16S RNA-seq. This is a Kinnex-based service, and also we offer full-length 18S and ITS RNA sequencing. So this is a mass-seq-based sequencing on PacBio. And we also offer a shotgun metagenomic profiling and assembly services as well. So just a little bit note here that at Novogene Europe, we'll actually also accept customer-built PacBio libraries, which is compatible with Sequel 2 and 2E and also Revio system for sequencing as well. So this is sequencing-only services. And also, starting in 2024 Q3, so Novogene Europe will also offer high molecular weight DNA extraction services at our German laboratories. And you're going to hear the more details about those technologies later in the presentation. Okay. So let me now introduce our special guest speakers for today's webinar. So the main speaker will be Dr. Thea Zieglar, who is a sequencing application sales specialist at PacBio. And Dr. Zieglar has extensive experience in sequencing applications and will be our main speakers today. And then another special guest today is Dr. Dimitar Petrov, who's a field application support scientist at PacBio. And Dr. Petrov will join us during the Q&A session and will provide additional insights and answer questions. Okay. So now, without further ado, let's get started. Please join me to welcome our main speaker, Dr. Thea Zieglar. Thank you. Okay, the floor is yours.
Thanks, Shi-qi. Thanks a lot, and welcome everyone. Just do you see my screen?
We do indeed, Thea. It looks good.
Perfect. Okay, then in the next minutes, I'm going to provide an overview of PacBio's technology and background, as well as of our most important applications. The work on PacBio's smart sequencing technology goes back over 20 years. The first commercial platform was then launched in 2011, the RS1 system. Its sequencing capacity was still quite limited, but thanks to the long read lengths, the RS1 already enabled researchers to read through difficult genomic regions, such as large repeats or complex structure. And since the release of the RS1, PacBio has scaled its sequencing capacity 1300-fold. So the newest Revio system enables the sequencing of large genomes and even several human genomes in parallel. You can sequence one human genome per smart cell on the Revio system, or up to 4 in parallel in only one day. In addition, the long-awaited goal of the $1,000 or euro long-read genome has finally been achieved. And with this new competitive pricing, PacBio sequencing finally got attractive for the broader research community. PacBio single-molecule real-time or smart sequencing technology involves the sequencing of single circular molecules on semiconductor chips. These chips have millions of tiny wells into which only one DNA molecule can bind at a time. Smart sequencing uses the natural process of DNA replication. So correct base integration is detected via fluorescently labeled bases. Nowadays, we talk about HiFi sequencing when we mean PacBio long-read sequencing. And HiFi stands for high fidelity, as the sequencing accuracy of a HiFi read is on average 99% or higher, and the technology is considered the most accurate among sequencing technologies. And the secret behind that is depicted on the right. HiFi reads are produced using circular consensus sequencing. And in order to generate such a circular DNA template, you will ligate 2 adapters on each end during library preparation. And this allows the polymerase to make several passes around it during sequencing. And thanks to the random distribution of errors, we can generate a highly accurate consensus sequence from the different subreads, which is our HiFi read. The superior performance of HiFi sequencing has been validated in many different technology comparisons. Here, we're looking at the results of one of the most recent ones from the All of Us initiatives. The All of Us initiative aims to sequence the genome of over 1 million Americans from diverse ethnic backgrounds to gain insights for personalized medicine. And in this technical pilot test, they compared the performance of traditional short-read sequencing with the long-read sequencing technologies. Their analysis revealed substantial differences, especially for complex medically relevant genes, particularly in terms of gene coverage and pathogenic variant detections. And while there are over 4,600 medically relevant genes in the human genome, 386 of them are known to be challenging, meaning that exome and whole genome sequencing approaches with short reads often fail to reveal pathogenic variants. And the results of the study show that HiFi sequencing outperformed the other technologies in both precision and recall for these challenging genes. And also overall, the results show that HiFi reads produce the most accurate results for both small and large variants. And as for large structure variants, you also see that short reads will usually miss more than half of these important variants. So what makes me think that structural variants are so important? As per definition, they are typically larger than 50 base pairs, and therefore they contribute to a greater diversity at the nucleotide level between 2 humans than small variants. And in addition, it has been shown that structural variants are responsible for 25 to 30% of all rare protein truncating events per genome, and 50% of them are pathogenic. And as you see in the figure on the right, this is a much larger proportion than for any other variant class, and therefore they should not be ignored in studies. HiFi sequencing has contributed to several important milestones in the field of genomics. You've probably heard about the 2 depicted ones, which were published in 2022 and 23. The first of them was the completion of the human genome, realized by the telomere-to-telomere consortium. In the former human genome reference, 8% of sequence were still missing, which could be finally resolved thanks to long-read technologies. HiFi sequencing builds the backbone of this new assembly, making up 99% of this final sequence. The project saw an outstanding consensus accuracy of Q72, which was also a result of PacBio's HiFi technology. The Human Pangenome Reference Consortium builds on this and is aiming to create a pangenome reference that reflects human diversity. And besides the involvement in these 2 important consortia from the human genomics field, HiFi sequencing has also been employed in many other consortia works for plant and animal genome research or biodiversity initiatives for the generation of reference genomes. So now we already looked into some of the paradigm shifts in the field of genomics, but besides this, we're also seeing further shifts in the fields of epigenomic and transcriptome research. In the epigenomics field, HiFi sequencing is able to detect epigenetic signatures through monitoring the polymerase kinetics during sequencing. The detection of bacterial markers such as 4-methylcytosine and N6-methyladenine has been possible for a long time. 5-methylcytosine, however, the most important methylation marker in vertebrates and human, was more difficult to call due to a weak kinetic signal. But thanks to the integration of a new deep learning algorithm to our sequencers, they've turned into 5-base sequencers, as methylation calling is done on the instrument. And this allows researchers now to shift their focus entirely on the biological or medical questions instead of worrying about the analysis. In the transcriptomics field, several innovations, both on the instrument and application levels, have led to a significantly increased throughput and decreased prices, which initiated a new era of single-cell transcriptomics. And we'll have a deeper look at this in the application section. A good way to illustrate the advantage of HiFi reads for research investigations or variant discovery is to compare it to a short-read genome. We know short-read whole genome sequencing is good in resolving SNVs and indels, but as you know and see here, there are many other variant types that short reads cannot resolve very well. We've already talked about structural variants and have seen that long reads usually unveil around 2.5 times more of them. But there's also other information that you gain with long reads. HiFi sequencers enable 5-methylcytosine methylation calling, as we've already touched on, together with the base calling. And this is without any additional library prep and cost, so just in the same sequencing run. Also, there's the advantage of resolving segmented applications, so highly identical regions in the genome, which are usually poorly covered by short reads, even in the gene coding regions. Another advantage is the phasing of different haplotypes. So instead of getting only one collapsed 3 gigabase genome, such as with short reads, you'll be able to resolve both the maternal and the paternal allele with HiFi sequencing. And all of these advantages come in addition to strong SNP and small indel calling, which is good or even better, as you've seen, as standard short read sequencing. One very important step in the generation of high-quality sequencing data is the purification of high-quality DNA. And in order to support our users with this critical step, PacBio has acquired Circulomics and its nanobind technology a few years ago, and is thus able to offer these kits for high-quality, high molecular weight DNA extraction, together with the protocols for various sample types. Also, our support team is always happy to provide assistance or answer any questions in case of challenging samples. So during the introduction, you have now already heard about the power of HiFi sequencing for genome assembly, variant detection, and also about the simplicity for the detection of epigenetic modifications. So we'll focus now more on targeted sequencing, RNA sequencing, metagenomic applications in the second part. As classical NGS technologies, HiFi sequencing is compatible with targeted sequencing methods. The two most common approaches are depicted here. Due to the longer read lengths, the complexity and effort of primer or probe design is significantly reduced in both cases. Typical amplicon lengths for HiFi sequencing are up to 10 kilobases, but there are also cases where users have gone further. So ultimately, anything that can be amplified can also be sequenced. Long-read amplicon sequencing is a method of choice for those interested in single genes or smaller gene panels. For larger panels, as a rule of thumb, we would say for more than 10 to 12 target genes, we'd recommend target enrichment as a method of choice. And as a suitable partner, we can recommend Twist Bioscience, who were the first to optimize their hybridization protocol specifically for HiFi sequencing. Both amplicon sequencing and target enrichment are well established and provide very robust results in combination with HiFi sequencing, even for regions that are not accessible with short reads, with the well-known advantages of long reads, such as for the resolution of different alleles. Another targeted sequencing solution that I'd like to introduce is our new Pure Target kit, which offers a solution for repeat expansion sequencing. Repeat expansions are traditionally very hard or not possible to resolve with short reads, and this is especially true for long repeat expansion genes, which are longer than the read lengths of short reads of around 250 base pairs. However, there are many repeat expansion genes involved in neurological disease, and determining the exact length is important for distinguishing between healthy and pathogenic variants. For this reason, PacBio has compiled a panel for the most important repeat expansions, including many ataxia genes and Fragile X, with a total number of 20 genes. The target enrichment approach is CRISPR-Cas-based and therefore does not require PCR amplification. The scalable and robust workflow enables a comprehensive characterization of these 20 hard-to-characterize targets in one single approach, and it takes only 3 days from DNA extraction to the analysis results. So for comprehensive genotyping of repeat expansions, it is important in many cases to resolve repeat sequence to a single nucleotide level. Here's an example for FMR1, where the repeat motif is primarily CGG, but we can clearly see an ATG interruption on both alleles. Thanks to the unique combination of long read lengths and single base accuracy, HiFi reads can deliver this information in one single assay. Comprehensive genotyping of the repeat expansions also requires the accurate sizing of expanded alleles. Having an accurate sizing for all allele lengths can help inform whether the repeat is pathogenic, and it can be predictive of disease severity and age of disease onset. And as you can see, the repeat sizing is phased, and we see the short allele at the top of the long allele below. And in addition, as a pure target approach does not require PCR amplification, you'll be able to obtain methylation information with this approach without the need for a separate assay. The last big advantage of our pure target approach is that thanks to the deep coverage that is generated, you're able to characterize mosaicism, or the variation in repeat lengths within a sample. High levels of mosaicism are associated with increase in repeat lengths in somatic and germline tissues, and being able to sequence deeply at mosaic targets is important for studies of disease mechanisms or drug research. So in summary, our new pure target repeat expansion panel offers an end-to-end solution with a convenient Cas9-based assay and a 20-gene panel. The pure target libraries sequenced with HiFi sequencing offer comprehensive genotypes, which include the accurate sizing of long repeat expansions, base resolved repeat sequence, methylation information, and a high read depth for mosaicism. We're now coming to our RNA sequencing applications. In recent years, it has been repeatedly shown that the importance of alternative splicing in different isoforms of genes has long been underestimated. And since different isoforms lead to different proteins, which represent the carriers of function, it is important to be able to make an accurate distinction. A popular example for a gene for which a distinction of the different isoforms can decide between life and death is BCL. One isoform of it promotes cancer, and the other suppresses it. Due to their fragmented nature, short reads are not well suited to distinguish and unveil different isoforms reliably. They are essentially trying to assemble a puzzle where there often is not a unique answer. And in contrast to that, long reads don't need to solve a puzzle, as they are able to sequence transcripts or the corresponding cDNA in one single piece. So there's no assembly of pieces required. Or could you tell here on the left side how many isoforms are there? PacBio's full-length RNA sequencing method IsoSeq has been a valuable tool for researchers to distinguish and characterize isoforms for a long time. However, the IsoSeq method has not been very cost-efficient in the past, which has limited its use. And this is because the average transcript length is between 1 to 3 kilobases, while the optimal read length of a HiFi read is 15 to 20 kilobases. And to solve this problem, researchers from the Broad Institute developed a method a few years ago that concatenates transcript cDNAs into fragments of an optimal length for HiFi sequencing. The method is very robust and provides the same sequencing quality as a classical iso-seq method, since all cDNAs are labeled with unique barcodes and delinked into individual transcript cDNAs in the end. And for this reason, PacBio has collaborated with the developers of the method and launched a new range of kits based on it, the Kinnex kits. For full-length RNA sequencing, the Kinnex approach enables a 12-fold concatenation, yielding up to 40 million reads on the Revio system. This is 60 times more data than a few years ago, before the Revio system came to the market and before the Kinnex kits were developed. And for this reason, RNA sequencing with PacBio is much more affordable than ever before, and the discrepancy to short read sequencing costs are no longer very large. And especially when you consider that a long read provides significantly more information than a short read, and significantly less sequencing depth is required. So with Kinnex full-length RNA, you can detect novel isoforms and variants with confidence. Plus, with 40 million reads per experiment for the first time, you're also able to do quantitative experiments with long reads alone. So no more short reads required for abundance information. This allows you also to characterize differential transcript usage and to predict their functional impact. If wished, multiplexing of different samples in one run is no problem, thanks to the different barcoding options. And also, not to forget, the Kinnex kit comes with an integrated solution for bioinformatic analysis, enabling isoform classification. With the increased throughput of the Kinnex method, it is possible for the first time to reach saturation for the number of detectable isoforms of a cell type in a single sequencing run. Our R&D team has sequenced different cell lines and tissues to make recommendations for the design of experiments. As you can see in the figure on the left, at 10 million reads, 80% of non-isoforms are detected. And reducing the sequencing depths to 5 million reads per sample can reduce the costs even more, while still capturing significant numbers of genes and isoforms. So this was shown here in the example of the pluripotent stem cell line, WTC11, where reducing the read depth from 10 to 5 million reads still detects about 90% of the isoforms. And this would allow you to run up to 8 samples in parallel on a Revio smart cell. This table provides a summary of our sequencing recommendations. So for isoform discovery in comparison of moderate to rare transcripts, we recommend a read depth of 10 million reads. Ooh. Sorry for that. So so for detection of isoforms with which are moderately expressed, we recommend a read depth of 10 million reads. And this would allow you to run up to 4 samples in parallel on a Revio. For isoform discovery of highly expressed transcripts, you can reduce read depths to 5 million reads, to 5 million reads, and this would allow still an 8-plex on a Revio. This can be really useful for biomarker discovery and biopharma applications, or for example, for the detection of fusion transcripts, which are usually quite high expressed. For comprehensive representation of expressed isoforms in plant or animal research, the choice of the read depth depends more on the complexity of the species genome and transcriptome. Therefore, we usually recommend to sequence different tissue types and to start with 5 million reads per tissue. If more read depth is required, another smart cell can still be run. In addition to the Kinnex full-length RNA kit, Kinnex is also available for single-cell RNA sequencing and 16S sequencing. You'll hear more about this later. The Kinnex single-cell RNA kit allows to investigate isoform expression at a cellular resolution. The kit is compatible with both the 10x chromium 3' kit and the chromium 5' kit. And thanks to a 16-fold concatenation factor, up to 100 million reads can be achieved on a Revio. This is sufficient for a typical single-cell experiment with 3,000 to 10,000 cells. Thanks to the barcoded Kinnex adapters, multiple single-cell experiments, or up to 4, can be sequenced per Revio run. The Kinnex single-cell RNA kit also comes with an integrated bioinformatic pipeline that is integrated to our SmartLink software that generates gene and isoform count matrices, which are compatible with classical tertiary analysis software for single-cell experiments. So here's an example from the cancer research field that impressively demonstrates the power of high-throughput full-length single-cell RNA sequencing. The study was carried out by Swiss researchers around Nico Berenwinkel and the Swiss Tumor Profiling Consortium, even before the Kinnex method came onto the market. However, the approach and the achievable number of reads per cell are identical to that of the Kinnex approach. The results of the study show that PacBio single sequencing provides an equivalent cell type classification as short-read single-cell sequencing, and on top of this, it allows the discovery of large amounts of novel isoforms. It also enables the detection of known and novel fusions, and even permits the accurate detection of cancer-related expressed mutations. We will have a deeper look at these features in the next slides. So to illustrate the significance of this new method in combination with HiFi single-cell sequencing, you see here that in the study with the three ovarian cancer patients, already 150,000 isoforms were detected, and 30% of these were new. And that is nearly 50,000 isoforms that were not previously known or had new splice junctions that were not represented in the catalog. In addition, the study showed the functional relevance of isoform-based analysis compared to the gene-based analysis of classical short-read approaches. Gene-based expression could not reveal differences in expression between healthy and cancer cells, whereas isoform-based analysis with long reads revealed differential isoform usage in different cell types, as in this example of IGF-1. However, several other genes also showed significant switches between healthy and cancerous cells. So IGF-1 was no exception. The study also showed that long-read sequencing reliably captures gene fusions, and even this IGF-2BP testbar fusion that was previously misidentified in short-read data. And due to the intrinsic limitations, short-read single-cell RNA sequencing often fails to distinguish between gene and fusion gene expression, potentially leading to wrong biological conclusions. So altogether, the study of Arthur Dondi and his colleagues show impressively that long-read sequencing provides a more complete picture of cancer-specific changes. Only highly accurate long reads can reveal the consequences of RNA dysregulation in cancer. And this is also true in other research contexts, where long reads have shown that isoform switches mediate transitions to help organisms to adapt to different environmental conditions or mediate transitions from healthy to disease states. Yeah, and with that, we're coming to our last application section, microbial genomics. This slide shows an overview of our microbial applications. HiFi sequencing makes it possible to sequence microbial genomes in excellent quality and to obtain single contig chromosomes for most bacteria. With the Revio system, up to 96 microbial genomes can be sequenced in parallel on only one smart cell, which makes the costs very attractive, even compared to short reads. And in addition to microbial whole genome sequencing, HiFi sequencing also offers great advantages for metagenomic applications. Full-length 16S sequencing enables a resolution down to the strain level that cannot be achieved with classical NGS. For projects with higher sample volumes, our Kinnex 16S kits offer an economical solution that allow up to 1,500 samples to be sequenced in parallel with only one Revio smart cell. Also, in shotgun metagenomic sequencing applications, HiFi reads offer significantly higher information content than short reads. On average, one HiFi read contains around 8 genes, and 90% of reads will have at least one gene. And this allows you to profile entire gene clusters with high precision and recall, for example, for the identification of new metabolic pathways. Similar advantages apply to shotgun metagenome assembly, where HiFi reads can be used to generate many high-quality metagenome assembled genomes, or MAGs, per sequencing run, and even resolve closely related strains. So this study, which was led by George Weinstock, the leader of the Human Microbiome Project, demonstrates really well that full-length 16S sequencing is the only way to resolve all bacterial clades in the human gut without bias. The gray panels depicted on the right show that the variable regions of the 16S operon defined by commonly used primer binding sites for short-read NGS taxonomic studies. And depending on which variable region is used, the proportion of bacterial taxa that cannot be identified at the species level varies significantly. Only full-length 16S sequencing can resolve all bacterial clades. For metagenome assemblies, the goal typically is to assemble a representative genome for each microorganism in a microbiome sample. However, there are several challenges that only accurate long reads can resolve. For example, there are multiple species present in a sample, leading to uneven and unknown coverages. And there are many repeat regions and intergenomic overlaps across species and strains, and these pose a hurdle to short reads that they usually fail to solve. In contrast to that, thanks to the combined advantage of long read lengths and high accuracy of HiFi reads, they allow to assemble a large number of high-quality MAGs in a single metagenomic shotgun experiment. Thanks to new bioinformatic methods, the yield of high-quality MAGs for HiFi metagenomic assembly has been increased even further. In addition to the tool HiFi as Meta, which we have been recommending for several years now, the HiFi community was enriched by another powerful tool, Meta MDBG, at the beginning of this year. The following example of genome-resolved metagenomics in wetland soil demonstrates the new abilities made possible by the high throughput of our Revio system and new bioinformatic methods. The project was a collaboration with Professor Jill Binfield from UC Berkeley and her team. Soil samples are usually so complex and rich in bacterial populations that they have been difficult to characterize in the past, especially since many of the bacteria are difficult to cultivate under laboratory conditions. For this project, 6 samples were sequenced in total, and 3 of them with a new Revio 25M smart cell, yielding 195 gigabase of total HiFi data. The assembly was realized with HiFi as Meta and Meta MDBG, and the binning and quality control was done with our HiFi MAG pipeline. In total, over 1,200 MAGs were assembled from the combined soil samples. And out of these, 45% were high-quality MAGs, meaning that these MAGs have a completeness of over 90%. About 1/3 of these MAGs was even single-contig, requiring no assembly at all. The identified MAGs represent over 320 genera, and around 550 of the metagenome assembled genomes could not be assigned at the genus level. And this is because a lot of species or genus level genomes from environmental habitats aren't in the reference databases yet, so the taxonomic calls can't be made at that level. And that's a big advantage of high-quality MAG assemblies with HiFi sequencing, to enable researchers to get more of those, to then be able to add them to the reference databases without needing to culture them first for single isolate whole genome sequencing. I hope the selected examples could show that the increased throughput of our Revio sequencer, as well as innovation at the application level, such as the new Kinnex kits, open up new application horizons and facilitate the access to long-read sequencing thanks to significantly reduced prices. This slide provides an overview of the sequencing capacity of our different applications in combination with the Revio system, which is available through Novogene. For example, if you're interested in whole genomes, one human genome, as an example, at 30-fold coverage could be sequenced per Revio Smart Cell. On the case of microbial genomes, up to 96 samples could be sequenced in parallel. In the field of targeted sequencing, there are hardly any limits with the high throughput of the Revio, so several thousand samples could be pooled per Smart Cell, which means that the per sample cost for large projects are extremely low. We have already discussed the recommendations for RNA sequencing applications earlier, and our recommendations for metagenomic applications shown here refer to human microbiome samples of known complexity. For environmental samples, the experimental design strongly depends on the complexity, and in soil it's usually much higher than in our gut. Thanks to the new Kinnex 16S kits, full-length 16S sequencing with long reads has become extremely cost-efficient, comparable to short-read costs. So I hope that the presented examples have shown you that HiFi sequencing provides a powerful new tool to interrogate living systems, allowing many new discoveries that were not accessible before. Because this would be very much in line with the vision of PacBio to enable the promise of genomics to better human health by providing the most accurate and complete view of genomes. Yeah, and with that, we're coming to an end, and we look forward to your questions. The Q&A session is about to start in a minute.
Right, thank you very much for the great talk. Let me just reshare my screen here. Okay. And Thea, would you mind to stop sharing, and then so I can put my screen on here. So basically in this session, okay, perfect. Let me just share screen. Sorry, why it's not working? Okay. Okay. So anyways, so in this bit, we actually have 3 experts from PacBio, and also myself from Novogene. And so please feel free to submit your questions during the Q&A session features, and then we'll do our best to address them. So if you don't get your question today, we will still follow up via email, probably sometime next week. Okay. So I start with some general question. Okay, so the first one is probably for Thea. So what specific exclusive advantages are there to detect structural variants with PacBio? And is it possible to combine or utilize the previous short-read data still?
So, of course, compared to short-read sequencing, the big advantage is that thanks to the long read lengths, HiFi reads are usually able to span structure variants and long repeats. So, yeah, this allows you to resolve them. And the second part of the question was if you can combine this with what? With
a short read.
Yeah, in theory, this is possible if you wanted to do that as a lower sequencing depth, that you could combine this data with short reads. But you also don't need this, because if you go a bit deeper, you're also able to resolve smaller variants.
Okay, thank you very much for that answer. And then next question is probably a little bit more for bioinformatics, so probably for David. Basically, so the question is that do we have to use a specific software for long-read sequencing, or can we still use some of the previous software that is used for short-read already?
Yeah, that's a very important consideration for the data analysis. So for long reads, for HiFi reads specifically, some of the tools that you might have used for short reads can be used, but typically you want to look at long read specific tools. That's very important to get the best performance. For example, for mapping, you need a different algorithm if you have a 10, 15, 20 KB read as compared to a short read. So as an example, Minimap2 is typically used, or the PacBio wrapper of that, PBMM2. And that continues to variant calling. So for structural variant calling, as Thea mentioned, the long reads, that's where they're really powerful to span large deletions, but especially also large insertions, inversions, translocations. And there you want to use a long read specific tool. And for HiFi reads, we recommend PBSV or Sniffles as very powerful tools for structural variants. For short reads, the best performance is observed with deep variant and the HiFi specific model. And then there are additional tools which can really show the power of the HiFi read. So as Thea explained, the tandem repeat resolution. So there's a specific tool for that, TRGT, it's called Target. Also for large CNVs, so for hundreds of kilobases to megabases, there's a tool called HiFi CNV. So in general, typically it's specific tools for HiFi reads that we recommend, and we have a GitHub repository where these tools are also summarized. I recommend to have a look there.
Okay, and thank you very much. That's a very comprehensive answer to that. And then the next question is probably more a wet lab question, so probably for Demeter. So basically, we have attendees wanted to know if that PacBio HiFi library preparation is also suitable for FFPE DNA or RNA samples. And also a follow-up question that usually, how long it takes generally for library preparation for PacBio HiFi libraries.
Thank you. Well, the normal, or actually, all the library preps, of course, start with double-stranded DNA. So this doesn't include the extraction of DNA. So basically, coming to the original. Question is also our own kit, for example, which is optimized for high molecular weight DNA extraction, which of course with samples where the DNA is degraded might not perform or deliver high molecular weight DNA, but still it would be long enough for HiFi sequencing. And then the library prep, once you've extracted your DNA, takes roughly 4 hours. And this would be independent, basically, what's the origin of DNA. It could be also amplicons, it could be also RNA converted to double-stranded cDNA. It will always take 4 hours, roughly. Yes, so, and those samples you can prepare as well.
Okay. Can I just follow up on that question then? Because, ideally it's high molecular weight, but, even as short as possible still can make into library. But what would be the idea, for example, size selection for PacBio Revio HiFi sequencing?
Yes, it's also a very relevant, important question, because of course the technology allows sequencing of different lengths of DNA, like very long or also shorter fragments, but we have an idea length of the quality and length is there at their optimum. And for that you need or we recommend size selection so that you have exactly those sizes and not also shorter ones, which reduce basically your final yield. We have 3 options which we recommend. One, like depending on the stringency that you look and the price that you're willing to go for. The more simple one would be a beat-based size selection, and then what we recommend is what we call a short read eliminator kit. This is a precipitation-based kit that we offer. It takes just like 1.5 to 2 hours to select for certain sizes, and then we have the best, let's say, in terms of performance, but in terms of size selection way, which is a geo-based size selection. So depending on the quality of sample, you can select which method you need.
Okay, of course. So, okay, thank you very much for that. And then there's another question, very interesting actually. So it's probably for David, I say. It's about Kinnex kit and for RNA sequencing. So basically the question is where the concatenation will introduce bias in a transcript profile, especially when it comes to convolution, and also how to get rid of those linkers sequence during the data analysis.
Yeah, great question. So the concatenation is random. So you expect that you add all sorts of transcripts and lengths together. So we're not aware of a bias. You see a nice distribution of read length. And then the second question about the linkers, or as we call them, the concatenation adapters, there is an integrated tool in the PacBio software that typically runs automatically or can run automatically, that separates the array, the segments, and the adapter sequences are automatically removed from there. So what you get at the end is a HiFi read file, which is already processed and that essentially contains exactly the reads as they would be without the concatenation.
Okay, thank you very much for that. And then I have a few questions actually all related to metagenomics sequencing actually. I think I'm going to ask this to Thea. So basically, the customer obviously has samples wanted to do microbiome research. And first question is about the depth that is required. Because when you presented, you have different, how many samples can put into one Revio cells. Could you just illustrate it a bit further? And what is the depth that's required for environmental samples for metagenomics sequencing?
So yeah, for metagenomic samples, the recommendations about the depths are really hard to provide as we don't know the complexity of the sample. So usually we say to resolve one stretch of sequence really well, we need 5 to 10 copies. But if you don't know how many organisms and species are present in the sample, this is hard to tell. However, as you've seen on the in the overview for our sequencing recommendations with the Revio, we can provide some guidance for human microbiome samples, as these have been sequenced quite often already with PacBio. So there you could sequence, for example, if your goal is metagenome assembly, up to 12 samples per Revio Smart Cell, and you could obtain at least 25 to 30 near complete metagenome assembled genomes out of that from the most dominant species in the samples.
Okay, thank you very much. And also related to this question is that for a sample of max, what level of the identification it can reach, the species or genus level, And also, if we do a shotgun long read metagenome sequencing, does it still require 16S, for example, for the taxonomy profiling?
So nowadays, I think David could answer this even better, but to my knowledge, there are enough HiFi adapted tools that allow the taxonomic identifications of species out of shotgun metagenomic profiling experiments. So you don't need any additional 16S sequencing. And what was the second part of the question, Shi-qi?
So the attendees basically still wondering, like, if they do the long read profiling, do they still need to have 16S, for example, in parallel for the analysis?
I think this was a part of the question that I already answered, but I forgot the first part of it.
The identification level, the genus or species or strain level for the max.
So in theory, it's really no problem to like resolve all sequenced organisms up to the species level. But of course, this also depends on the reference databases. And for environmental samples, there are still there's still significant gaps. So it's sometimes a little bit hard to call them directly, but like accurately sequence metagenome assembled genomes with one single approach and to fill these reference databases, especially for species that are hard to culture under laboratory conditions.
Okay, thank you very much. And David, do you have anything to add to that? And also that, and also, another question, if you can answer as well, is that can we get methylation information, during the metagenomics sequencing as well?
Yeah, that's a great question. And actually the beauty of the HiFi reads is that you get the methylation information, 5MC information on a read level, so on a molecule level. So indeed, also for metagenomic sequencing, and that's shotgun metagenomic sequencing, not 16S, of course, as you have amplification there. But for shotgun metagenomic sequencing, you get each molecule sequenced, and it does get a kinetic profile of the sequencing. So you do get methylation information for each molecule, meaning you can, for a population, for a metagenomic population, you can actually use methylation information if you want. Most of the tools, most of the pipelines don't include that at the moment. We're mostly looking at the sequence composition and establishing metagenomic assembled contigs, but you could add that information. I think there's great potential to look at that as well.
Okay.
And then for the 16S part, just before, you can actually extract the 16S sequence typically from assembled contigs, so there is no need to do additional 16S sequencing in most cases.
Okay, so if the researchers would just still want to look into the 16S, they can actually just directly extract from what they already got from the shotgun data. Okay, thank you very much. And then there's another question for the target region sequencing pure target. So the attendees want to ask, is there a size limit for the long read sequencing regarding target capture? And Thea, okay.
No, I think David can answer this question best.
Okay, maybe David, back to you. Yeah.
I'm sorry, could you repeat the question? I missed the middle part of it.
So for the targeted sequencing, is there any limitation of the size for the target capturing for HiFi sequencing?
That's mostly, so the size of a panel, for example, is mostly a question of how many samples you want to multiplex in a run. So it's a function of number of samples and the size of it. That would probably need to be discussed on a project level best.
Okay. So due to the time limitation, we're going to have one more question. So basically, the attendee says she's working with the worms that genomes is about 1 GB in size, and how many genomes they can sequence on one a smart cell. So David, since you are here, and maybe you can also answer that question as well. Yeah.
I wonder if Dimitar or Thea wanted to start with that, and I can maybe add on the analysis side.
Mm-hmm.
Yeah, so maybe if I understood correctly, the genome of the worm has approximately 1 gigabase size. So then you can you could probably multiplex around 3 of these genomes in parallel if you want. So depending, but one important question to clear before that would also be the ploidy level of the worm. So yeah, if it's a diploid genome, you could sequence up to 3 genomes.
Depends also basically on the coverage, actually. I mean, you can sequence even more. It would be for 30x, I would just complement Thea's response. If you're looking for 30x coverage, it would be.
Mm-hmm. Yeah. David, do you have anything to add on the answer?
No, actually, that was pretty complete. So thank you.
Okay. So thank you very much, everyone. And and I also thank all the attendees as well. Hope you found the webinar insightful and useful. And also special thanks for all the speakers. And as I mentioned earlier, if you haven't managed to answer all the live questions, then we will get back to you via emails next week with the answer. And we will also email you a link to the webinar recordings and the slide deck as well. So keep an eye on your inbox. When the webinar finishes, there will be a survey that will be pop-up. So we'd really appreciate that you taking the time to complete, as it will help us to make sure in the future our webinars are even more relevant to you and your research goals. So additionally, there will be a link that will basically follow up the email for you to review, and you will also obtain an attendee certificate if you need one. Okay, and in the meanwhile, please feel free to get in touch on social media, visit our website as well. So thank you again for attending today's webinar. Thank you.